There we go.
Okay, so the next slide was this picture here, and it asked you which one would be more efficient in removing waste by diffusion, and that's the smaller cell, all right, cell A. Okay, and then this question is asking you to look at these pictures and think about which one would have to be more efficient in transporting across the membrane. It actually should be cell 2, because cell 2, the cell membrane indents like this, they call it microvilli, which increases the surface area compared to the volume. So usually whenever you see that in a cell, it has a pretty high surface area to volume ratio. Cells that have microvilli like this are usually cells that their job is to get things into or out of the cell, either secrete things or take things in. So that's cell 2. The same idea here with the root hairs. This is a root tip, they call it. It's the end of a root. And the root cells, some of them are specialized where they have this protrusion out. That's called root hairs. That also increases the surface area and allows them to absorb more water than if they just were flat on the surface and didn't have the root hairs. All right? And then lastly, the number 6 had you calculate the surface area to volume ratio of a sphere. So the surface area is calculated 4 pi r squared. So it tells you the radius is 6, so you plug 6 in there and we get 452.16 centimeters squared. And then the volume is 4/3 pi r cubed, and so you plug in 6 and you get 904.32. Divide those and you should get 25. Okay? And so, like I said, I do have a surface area to volume ratio, and you're going to be given exactly, like I said, what is on this equation, this sheet here. All right. So then the second thing I passed back to you was the lab, the dialysis tubing lab. So if you get that out. I just want to hit on a few things. Most people are okay with writing the hypothesis, making your data table, doing the calculations, and showing the calculations. I do want to look at a couple of the questions. The first one is on the osmosis challenge sheet on the back side. It asks you to do the same experiment on the front side that we did together during class. I went over it together where they had these students had these different bags. They had this distilled water, 0.2 molar, 0.4 molar, 0.6, 0.8, and 1. And so in the example we went over in class, the outside solution in each one of these was distilled water. So the question at the back just is the exact same scenario, except instead of being distilled water on the outside, it's a 0.4 molar sucrose solution. So there's a 0.4 molar. And remember, the molarity is telling you the concentration of sucrose in that particular solution. And so we know that sucrose can't get through the membrane of the dialysis tubing. So sucrose is stuck where it's at. And so the only thing really then that can move in any of these situations or diffuse is the water. And so when you have a bag filled with distilled water in a 0.4 molar sucrose solution, water is going to move where? Into or out? Out, right? From a high to a low concentration. And so water moves out. For the next one, you have 0.2 molar inside and 0.4 molar outside. Where's your higher water concentration? Inside. So water will move out and the bag will lose mass. In this one here, this is an isotonic equilibrium state, so therefore water is going to move in and out equally. We will not see much of a percent change in mass. And then here we have the 0.4 molar solution therefore has a greater water concentration, so here we're going to see water start going in. So that's what we're going to see for this one as well as this one. And so we would see in which one would have the biggest percent change in mass. Would it be the 0.6 or the 1.0? Which one would have the biggest percent change in mass, do you think? I wanted to go over. And then the second one, most of it is in the questions. There were 10 questions that you had to go answer. Most of those questions, I kind of went over when we went over the actual lab, the day after we actually did it. But I wanted to look at, I think it's number 8. Is number 8 the one about starch? Double check my memory on that. All right, so in that one, I want to go over this because some people got it wrong and a lot of people asked questions about this before we turned it in. So it's saying that if we did the same lab that we did in class, we had distilled water on the outside, and if you had a starch solution in the inside of your dialysis tubing, what's going to happen? So what you have to do is think about, well, what do I know about starch? Well, starch is a polysaccharide, which means what? It's a long chain of, you guys remember, glucose molecules? So it's glucose, hooked to glucose, hooked to glucose, dot, 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 lots of glucose molecules. So I had some people ask me before they turned in the lab, well, how do I know if starch can get through the dialysis tubing? Well, dialysis tubing is selectively permeable based upon size. So we know that starch is pretty big, but we also know, can sucrose get through the membrane? No, sucrose cannot get through the dialysis tube, and we did that in the lab. Sucrose is stuck where it's at. Sucrose is just a measly little disaccharide. It's only 2 monosaccharides hooked together. Starch is many monosaccharides. So if glucose can't get through the membrane, guess what? Starch can't get through it either. It's way bigger. And so therefore, starch is stuck where it's at. So the only thing that you need is water. Where is water going to go? Into it. All right? So that's the only other one that I wanted to go over that I saw people have issues. Okay? All right. So the next thing I want to do is I want to talk a minute about your lab. Get out your lab and your graph for a second, and then we'll have some question and answer period for the chapter. Lab is due on Thursday, so you have some time if you have any other questions as they come up. But I just wanted to point out and go over with you how you're going to use your graph. So you have a graph set up something like this, where this is the molarity. So that's the molarity, and then you had 0 here. This is your percent change in mass. This is a positive, negative percent change. And so what you're going to do is, what we can do with this graph is figure out this question. What is the water potential of a potato? And so I'm going to show you how to use your graph to figure that out. So you made a line, you have your data plotted, and you already have a line. I'm just gonna arbitrarily draw a line. This line I just drew up here has nothing to do with what you should get. All right, it's just an example. So, where the line crosses the x-axis is of importance here. And so this is where there's 0% change in mass. What does that mean? Well, it means that this is what's true. This is the molarity of the outside solution when the potatoes are in equilibrium with the solution. Because when you get a 0% change in mass, there's not going to be overwhelmingly water going in or water going out, and so therefore it's in equilibrium. So, so what is that solution? And so when you take that 0% change, where that's at, you come down here, all right, and see what molarity is that. So I'm just going to estimate, let's say it's about 0.25. Here's like 0.3, all right. So for my example, that's a 0.25 molar solution. And so this is why the graph is important, because unless you got a 0% change for one of these numbers, you don't know, you just know it's between two numbers. So the purpose of the graph is to kind of give you an estimate of what molarity that might be. So I know that my potato then would be in equilibrium at a 0.25 molar solution. So what I need to do then is figure out the water potential of the outside solution. Water potential is the solute potential plus the pressure potential. If I can figure out the water potential of the outside solution, do I know the water potential of the potato? Yes, because they're in equilibrium. And so we have enough information to do that. So we don't know the solute potential right now. Do I know the pressure potential of that outside solution? It's exposed to the atmosphere. So it's 0, right? That's 0. And so, therefore, if I can figure out the solute potential of that liquid, then I can figure out the water potential. So how do I figure out the solute potential? In comes that second equation, negative I, C, R, T. So when I do this, this is a sucrose solution we were working with. So I is 1. And then C is the molar concentration. So at equilibrium, it would be 0.25 molar concentration. Your number is going to be where your line crosses. All right, so don't put 0.25, all right, because I'm putting it here. All right, so there's that. R, for simplistic sake, I'm not going to put the units. You can put the units, but it's going to be 0.831. And then the temperature, when we were doing the lab, I have a thermometer right there by that door, and it was 20 degrees Celsius, so I take 20 and add what? 273 to make it the units Kelvin. Multiply all this together, you're going to get some kind of negative number. I don't know what that is off the top of my head when I do that, but what I can do then is plug it in here and know the water potential of the surrounding solution that if the cells are in equilibrium, and then I know the water potential of the potato. Does that make sense? So therefore for you, all of these are constant. The only thing you needed to know here is the C, which is wherever it crosses the x-axis where it is equilibrium. And the only other thing that you needed to know was the temperature to be able to look at this. Does that make sense? Okay. And so that is what I want to point out about the lab. The lab's not due till Thursday, but that's how you figure out the water potential. So it's wherever it crosses that x-axis, you just plug it into our current equations. All right, so that's what I want to talk about with that. So I made that video and sent out the answer key for the water potential problems. And so if you still have questions about that, you can ask for about those today, as well as if you have your review sheet for chapter 6 and 7. What I'll do is just questions, all right? And we'll kind of go, we'll do like we did last time by chapter, all right? So we're not bouncing all over the place and switching gears all the time. So let me get that out. So if you do have questions about the water potential, we'll stick that with chapter 7 questions, okay? I will tell you I do have a water potential problem on the test, all right? So you don't have to memorize the equations, all right, or anything like that, or all the constants or anything like that, but you do have to tell me, okay? All right? Okay, you guys, so let's do chapter 6 first. So chapter 6 about a cell parts, basically, I call them.
Can we do number 6?
Number 6, not 6, yes. All right. This has been asked every hour, so this is
Yeah.
all right. Actually, no, I take that back. It wasn't asked in the second hour. Number 6, it gives you some information about some algae. It says a prokaryotic algae. So prokaryotic, it has no organelles, was recently discovered. This algae contains chlorophyll A and chlorophyll B. No other prokaryotic organisms contain both of these photosynthetic pigments. So these are things involved in the process of photosynthesis. But all photosynthetic eukaryotes, which means the cells have a nucleus and membrane-bound organelles and so on, do contain chlorophyll A and chlorophyll B. It says, explain why Lynn Margulis and other supporters of the endosymbiotic hypothesis were happy when they heard of this discovery. So to even answer this, you have to understand what the endosymbiotic hypothesis or theory is all about. So let me just review that with you. So what she said is that that there were prokaryotic cells on Earth. The fossil record shows that prokaryotic cells were first found on Earth and then eukaryotic cells. So her hypothesis is bridging the conversion of prokaryotic cell to eukaryotic cell. And so what her hypothesis states is this prokaryotic cell engulfed another prokaryotic cell, and that prokaryotic cell will later become the mitochondria. So how it engulfed that is by indenting the membrane like this. Like this. What process is that? It's not. Starts with an E. Oh, I think you're. What did you say? Phagocytosis, which is a type of the other one that starts with an E. Endocytosis. It's a way to get things in. Yes. All right. So now we have this becoming the mitochondria, an organelle inside the cell. Then she said that some of these guys here, there is another prokaryotic cell. So there's another prokaryotic cell that's going to later become the chloroplast, and it was engulfed the same way. So it engulfed like this. Here's your chloroplast, and then eventually we have a cell with the mitochondria and the chloroplast. All right. So her theory was that both the mitochondria and the chloroplast were once free-living organisms that were taken in and became helpful for the cell, and they became organelles. So that's the theory. So what she said is that she found some algae. They found some algae that had some of the pigments in it. The algae was prokaryotic. It doesn't have a nucleus or any organelles, but it contains pigments that allow it to do photosynthesis, chlorophyll, like the chloroplast. The chloroplast has my green marker here. The chloroplast has chlorophyll in it. So what she found is this algae that is prokaryotic, no organelles, but also contains chlorophyll. And so she said, well, there's these prokaryotic organisms now that don't have organelles and they contain chlorophyll and can do photosynthesis, which is exactly what she said the chloroplast originally was. The chloroplast originally was a prokaryotic cell that contains chlorophyll and can do photosynthesis and became the chloroplast inside of the plant cells. And so therefore that was proof that might have happened actually, that there are actually living cells that are prokaryotic that have chlorophyll and do photosynthesis without the chloroplast. Does that make sense?
Yes.
All right, other chapter 6 questions. Yes, Tom.
So, in the terms, there are plastics and peroximers.
Oh yeah, the peroxisomes in the second column underneath eukaryotic cells. I forgot to delete that out. I deleted it out of the list in question number 1, but I forgot to delete it out of the vocab. All right, so you don't have to know what peroxisomes are. Plastids that was in the first column, the third one down. Plastids are a group of organelles, but the one that you need to know is the chloroplast. Chloroplast is an example of that. Yes. Number 2, the importance of membranes and compartmentalization to cells. So what's the key for having membrane-bound organelles? Like basically it's asking you, why is it helpful to have a nuclear envelope, a membrane around the nucleus and around the mitochondria and the chloroplast and the Golgi apparatus and so on and so forth. So we have all these little different compartments in the cell. what's the benefit of that? You know?
Maybe so they can all like conduct their functions without like interference with the other organelles and getting them so they all have like their own space and like boundaries working.
Yeah, absolutely, absolutely. And so the enzymes and things that help those processes occur in each one of those would be concentrated in that particular area. So that's what I was getting at there. Other questions for chapter 6? Should we go on to 7? Let's go on to 7. If we have time at the end, if you have another chapter 6 question, you can come back to it. All right, so let's do 7. 7 is all about the cell membrane and allowing things into and out of the cell.
For number 6, what is a membrane pump?
Oh, okay, so for number 6, a membrane pump, the example that I went over is the Sodium-Potassium Pump. All right, so let's just kind of go over that. And there's a couple of questions with it. So that's the end of number 6 asks about membrane pumps, and then number 8 also talks about the Sodium-Potassium Pump. So let's kind of talk about those two together, because somebody's asked about the Sodium-Potassium Pump every hour here. So a membrane pump there's the lipid bilayer. You have proteins embedded in the lipid bilayer that molecules can move through, and the molecules that go through a pump, if this is a pump here, what that means is molecules will go from a low to a high concentration, and therefore will go what's called against the concentration gradient from against what normally happens. And so it'll go from low to high. So a pump, whenever we see the word pump, usually means it requires energy, right? What's the term for the type of transport that requires energy? Yeah, this is active transport. And when it requires energy, what's the form of energy that is usually used? ATP. And it requires energy because, like I said, it goes against the concentration gradient from low to high. So an example of a membrane pump that we talked about is the Sodium-Potassium Pump. And so let's just go through that here. If this is a cell membrane, and here's the Sodium-Potassium Pump. So it's clearly going to pump sodium and potassium, hence the name. Does anybody remember what it pumps out? Tom?
Free NaC2, free Na+.
Yeah, it pumps out 3 sodium ions, 3 Na+. And on the flip side, it pumps in potassium, and specifically 2 potassium. And this is a pump. It requires energy, which means these ions are going from a what to a what concentration? Low to high, because this is active transport. That's why it requires energy. So therefore, your high concentration of sodium is where? Into or out, in or out of the cell? Out. It's going to go from low sodium concentration to high sodium concentration. And then the opposite will be true for the potassium. So the potassium is being pumped in from low to high, so its high concentration is inside, and its low concentration is outside. And so, that energy is used to do this is in the form of ATP. So a real life example about why this is important is in neurons. Our neurons are part of our nervous system. Our neurons they depend upon always on the outside of the cell having a high sodium concentration, and on the inside of the cell a high potassium concentration. So therefore, it's worth it for the cell to expend energy to pump the sodiums out, to always have a high sodium concentration on the outside, and pump potassiums in, so you have a really high potassium concentration on the inside. If those ions reached equilibrium, the neurons would not be able to function, and if the neurons aren't functioning, then they're what stimulate our muscles to contract, and so on, and so organisms would die. So the neurons, this is important, they require a high sodium outside, I should say concentration, outside the cell, and let's do high potassium inside. And so it's worth for the cell to expend energy to do that, so that the neuron can continue to function. And then one other thing with this is when it does this, it creates a charge on the inside compared to the outside of the cell. The inside of the cell is slightly what? Yeah, more negative. Do you see where 3 positives are leaving and only 2 are coming in? So the inside of the cell, I'll do the third color here, is slightly more negative than the outside of the cell. All right, it's more negative than the outside, relative. All right, so that's the sodium-potassium pump. So that's like kind of 6 and 8 kind of together. Other questions? Yes.
Can we do number 11?
Number 11. Okay. So number 11, this is more of an analogy and kind of leading to the next chapter. Explain why a concentration gradient across the membrane represents potential energy. And so, let me erase this here. So if you have a cell and you have a concentration gradient, let's say there's an increased concentration, higher concentration of glucose on the outside of the cell than the inside of the cell. All right, first of all, where would glucose move if it can get through the membrane? Go into or out of the cell? Into the cell, right? High to low concentration. So glucose is going to go into the cell. This is the concentration gradient. A concentration gradient is the difference in concentration of a substance across the space. So it could be across the room, it could be across the cell membrane like this. So you have high, low, we say that there's a gradient, there's a difference in concentration. So it will move from, in this case, into the cell if it can. Let me ask you this. I'll get to the question 11 here, but this leads me to another question here. How is glucose going to get into the cell? When it goes through the membrane, what part of the membrane is glucose going to go through? Think you know, Charlie?
Transport protein?
It is a transport protein. How do you know? All right, it's too big to go through the membrane. So if we just did active, this was active transport. What's the opposite of active transport? Passive, right? So there were different ways that things can move in passive transport. The first way is through the lipid bilayer, all right? The second way was through a protein, and that's what Charlie just said, through a transport protein. There were 2 kinds of transport proteins. What are the 2 different kinds of transport proteins? Again?
Carrier and channel.
Carrier and channel. So channel has a tunnel kind of through it. All right, so these are channel proteins. And so some molecules will move through there. And then we had a carrier protein. Go on another room here. We have a carrier protein. Let me move this over here. Where this kind of changes shape, All right, I'm not the best artist here. All right, kind of changes shape like this. Looks like this as these bonding sites where molecules can come and bind, and then it changes shape and would allow it to go through that way. All right, so that's a carrier protein. And so Charlie said that it would go through one of these two things. All right, these are called facilitated diffusion, because what's facilitating the diffusion process? Proteins, right? Transport proteins. And this is just regular old diffusion, simple diffusion, and all of these collectively are passive transport, which means that you're going from high to low in all of these cases. So glucose, could you tell, so glucose, what has to be true about molecules that enter or leave a cell through one of these two things? Channel or carrier, what kind of molecules? What's true about molecule X here for these two? They have to be what? You guys remember the category of molecules that can diffuse, that need a channel protein?
A large uncharged polar molecule.
Large uncharged polar molecule
Or an ion.
or an ion or water. Whereas the ones that go through the lipid bilayer are what?
Small.
Small uncharged polar molecules or nonpolar. And the exception to the small uncharged polar was the water. All right, so glucose fits into the large uncharged, that's supposed to have a G there, polar category. So therefore glucose will go through a transport protein. All right, so I digress from the original question. So the original question was then going back to number 11 is that you have a concentration gradient, and how does this represent potential energy? So you have a bunch of glucose on the outside of the cell, and it's going to diffuse down its concentration gradient to a low concentration. So kind of like an analogy with water behind a dam, you have a bunch of water behind the dam. When it goes to the area of low concentration, flows down the dam, it releases energy that can be used to do work for us. The same kind of idea here, the glucose, the high concentration is like that water behind the dam, has a high concentration flowing to a low concentration, so it represents stored energy. Okay, so number 10 is about the carbohydrates. So I have on the protein. So I'm going to add to this particular drawing here. So on either a protein or a lipid, you can have a carbohydrate chain. So I'm going to put here, right here, this a carbohydrate chain where this is individual sugars hooked together to make a small carbohydrate chain. So when you have it coming off, if that's attached to a protein, they call that a glycoprotein. So this whole thing, the protein with the carbohydrate chain would be a glycoprotein. Sometimes those carbohydrate chains can be attached off of a lipid, and so that would be a glycolipid. And so those carbohydrate chains can come off of one of those two molecules, and the carbohydrate chain's purpose is are for recognition, so cell-to-cell recognition. So for instance, one cell, what makes it different is the type of monosaccharides that are hooked together and how long that carbohydrate chain is. So therefore, some molecules, some cells will have a different length and different monosaccharides that are hooked together than other cells. And so that's how one cell can tell the difference between, to know if it's the same type of cell or different cells, is if it's looking at that, kind of like I used the analogy of ID tags. So these guys here are different, the types of carbohydrates are different from cell to cell. And so one cell that has one type of carbohydrate chain, when it comes into contact with a cell that has a different carbohydrate chain, that's how it tells that it's not the same type of cell. Does that make sense? other questions? Gino?
Go with question 7.
7. All right. Simple, facilitated, active transport, and the significance of these processes to the life of the cell. So, perfect. So this is, I'm glad I left this up here. So, this, any type of diffusion, I just went over this, but I can go over it real quickly again. All right. I wasn't gonna say a word, Gino. I was just gonna go with it. All right. So this, when it moves, anytime we see diffusion after it, it means it's passive transport going from high to low, no energy. Simple, I didn't write this up here, but simple diffusion is when it goes from high to low through the lipid bilayer. Facilitated diffusion is going high to low through a protein. It can be either a channel or a carrier, it can be any type of protein, but the protein is facilitating or helping the molecule move through the membrane. Versus, versus active transport, which is with the pump, and that is going from low to high, requires energy. So the example of why that's important in real life is the neuron example. And then these guys here, having proteins embedded in the membrane, this is what helps to allow the cell to be selectively permeable. So it's selectively permeable about what can go in and out of it based upon charge and size. And so like a cell that might need, like I used the example with glucose here. If you had a cell that didn't have the transport proteins to allow glucose through, then guess what? Glucose doesn't get through the membrane. So therefore, having the appropriate types of proteins in there is what allows what can go in and out of the cell. So the reason why a cell can get glucose into it is because it has a protein in it that allows glucose to go through. And so therefore there's other channels that allow other molecules to go through, like for example the aquaporin allows water to go through high to low concentration and allow it to go in, and so on. And so what types of proteins allow it to be selectively permeable to different things. All right, that's why it's important. All right. Plasmolysis. All right, so this is what only happens in plant cells. And so let's just review a plant cell here. The outermost part of a plant cell is cell wall, right? And then underneath the cell wall is the cell membrane. And then the largest thing in the cell that's this big storage is the central vacuole here. So this stores water, pigments, poisons, all kinds of things here. So you have water storage here, and then this is the cytoplasm out here, which also, remember, the cytoplasm is a high concentration of water as well. And so what happens is when plasmolysis occurs, what kind of solution does the cell have to be in for this to occur? For those, do they know? For plasmolysis to occur, does water have to go into or out of the cell? I remember. I know the question is about what plasmolysis is, but I'm trying to see for the class if you like. Is water going to go into or out of the cell? Think you know?
Plasmolysis is when it goes out.
Yeah, water is going to go out of the cell. From the central vacuole, it also leaves from the cytoplasm, and so water leaves the cell. What's true about the solution on the outside of the cell if water leaves? It's called a hypertonic solution. What's true about a hypertonic? What's true about water concentration and solute concentration? Is water concentration higher in or out of the cell? In. So it has a lower concentration outside, higher water concentration inside, hence water is leaving, which means that the outside has a higher solute concentration, which gives it a lower water concentration. The more solutes you have, the lower the water concentration. So water is going to go out. So what is plasmolysis? So now that we understand here water is leaving, what happens is, remember plant cells, when they're filled with water, that let's get a different color here, that when it's filled with water, it puts pressure on the cell membrane and it butts up against the cell wall, but it doesn't explode because the cell wall resists that pressure. And so therefore, that's the best solution for plant cells to be in. But what happens is when you put it in a hypertonic solution and water begins to leave, guess what? That pressure starts going away, and what actually it would look like if I were to redraw it over here is that the cell, my cell wall is still there, but the cell membrane indents away from the wall, like this. All right, and because there's less water inside, putting pressure, pushing that cell membrane against the cell wall. Does that make sense? And that process is called plasmolysis. So it's the membrane kind of shrinking away from the cell wall because there's less water in there putting pressure and pushing it against it.
What would happen if a plant cell was in an isotonic?
In an isotonic solution, it would go in and out equally, but it wouldn't stand up. It'd probably be kind of wilty because it doesn't have that enough pressure to stand up straight. Okay. Any other questions? Megan. What is what?
It's on the vocab sheet.
Lysis? Oh, that means to like split or break. So when lysis occurs, if you do the same thing, if you had to put an animal cell in the same situation in water, or not in the same situation, in a situation where water goes in and it balloons up and explodes, we'd say the cell has lysed, all right, or it has split or broken open, okay? Other questions?